guys, welcome to Bio 112 Online, your home for biology education. Today's topic, the immune system. When we refer to the immune system, of course, we're referring to an animal system of defense against pathogens. We're defining pathogens here as infectious agents that cause disease. And we could break this immune system into two main units, the innate immune system and the acquired immune system. The innate immunity is a system of defenses that are active immediately and are not geared towards any specific pathogen. However, the acquired immune system is going to be a system of defenses that are slower to develop, but are able to detect and target a specific pathogen. The acquired immunity is only found in vertebrates. It has specificity, memory, and self-non-self recognition. It is slower to respond than the innate immune system, but once it gets going, it has two advantages, one of which is that it's tremendously efficient at targeting specific pathogens, and second, it has memory, which means that on the second exposure, it has an even enhanced protection. This acquired immunity can be broken up into two main segments, the humoral and the cell-mediated responses. Humoral immunity will secrete antibodies, complement proteins, and antimicrobial peptides in response to an antigen. The cell-mediated immunity is going to be the activation of antigenic-specific cytotoxic T cells, or phagocytes, and the release of cytokines in response to those pathogens. So in general, the acquired immunity is what we're going to be talking about today. Recall when we were talking about the innate immunity, we said that there were 5 main types of white blood cells: neutrophils, eosinophils, basophils, lymphocytes, and monocytes. For today, we're going to focus only on the lymphocytes. Those lymphocytes are found within the lymphatic system predominantly. The lymphatic system is going to be a system of vessels and nodes that transport interstitial fluids and houses those immune cells. We can envision this, the interstitial fluids, as all of those fluids that are surrounding the cells. The lymphatic system will go along and will collect those fluids and transport them through a network of vessels into a series of nodes. As they move their way through those nodes and through those vessels, they eventually find their way, i.e. the fluid, finds its way back to the blood. Along the way, you have the series of places where those lymph vessels will collect into central nodes. The major ones in our body, of course, happen right here, underneath our jaw, underneath our armpits, in the spleen, in the thymus, and in the groin area, along with some smaller nodes located throughout the body. These lymph nodes are going to be locations where many of the lymphocytes are found. Lymphocytes are going to be a group of mononuclear leukocytes housed in the lymphatic system and are responsible for acquired immunity. By mononuclear leukocytes, we're essentially meaning that they are white blood cells with a single large nucleus, like this one. The lymphocytes originate from stem cells in the bone marrow, much like most other blood-borne cells. Those that stay and fully develop within the bone marrow are referred to as the B cells. A subset depart the bone marrow prior to full development and make their way to the thymus. As they emerge from the thymus, they're referred to as the T cells. So think B for bone, T for thymus. The B cells are going to be lymphocytes that produce and secrete antibodies, while the T cells can be broken up into 2 main classes. The first of these are going to be the helper T cells. These are going to be lymphocytes that carry the CD4 receptor, and they act to stimulate the activity and growth of other lymphocytes, both B and T varieties. The other main type of T cells are going to be the cytotoxic or killer T cells. These are lymphocytes that carry a different receptor, the CD8 receptor, that act to destroy body cells that are either malfunctioning or harboring bacteria or viruses. As we go through and talk about these, along the way we're going to have a variety of different types of cell membrane bound proteins. Each of these receptors are going to deal with an antigen. An antigen is going to be any foreign molecule that is recognized by those lymphocytes and which elicits some type of response. On the antigen, there's going to be a specific place that the receptors or other proteins are going to bind onto. That place where they bind onto is referred to as an epitope, or a small accessible portion of the antigen that the lymphocyte actually recognizes and binds onto. So we're going to have four main types of proteins that we want to talk about today. The first of these are the major histocompatibility complex class 1 molecules. These are going to be found on nearly all of your nucleated cells and will present an antigen signal if they are infected. Most of the time, as your cells are going through and doing their normal processes, every so often they will take small pieces of what they're doing, of the proteins that they're producing, and they will put these onto the MHC1 molecules. They will then display these on their cell surfaces as a way of showing what they are doing and what's going on inside them. Cytotoxic T cells will then come through and read these to ensure what's going on inside the cell is what's supposed to be going on inside the cell. If tumor cells, in some cases, lack these MHC1 molecules, this absence is detected by the NK cells, the natural killer cells that we described in the previous video. These MHC1 molecules will have a domain that maintains itself within the cell membrane and several domains that will go through and stick above and outside of that membrane. They would generally house an antigen or some other piece of protein between their two alpha domains. These are similar to the major histocompatibility class 2 molecules. These MHC2 molecules are found only in antigen-presenting cells. These include the dendritic cells, the macrophages, and B cells. They will go along and they will display these antigens to helper T cells. When they do so, they help activate those helper T cells and will increase the response of the immune system. So the MHC2 are a dimer with two main proteins which emerge from the cell membrane and then each have a series of complexes that attach to the antigen. Our third type of receptor is going to be the B cell receptor. This is going to be 4 polypeptide chains that are linked by disulfide bridges. They are found only on B cells and have constant regions that do not change. But then they do have small regions that are referred to as the variable regions. These variable regions will differ between each individual B cell. For the B cell receptor, it looks kind of like a Y as you fit those 4 protein subunits together. You have those 4 variable regions. Each of those variable regions will then be the location where the antigen sits. In addition, you have a couple extra proteins, the CD79 proteins, which will go through and act as ways of regulating signal transduction and initiating signal transduction should the receptor be activated. Now, these B cell receptors are interesting because they act not only as a receptor, but they also play a second major role. They become antibodies or immunoglobulins. These antibodies are going to be the soluble form of the B cell antigen receptor, i.e., they can detach from the B cell and become their own effective portion of the immune system. Our final type of receptor that we wanted to talk about today are going to be the T cell receptors. These are going to be two polypeptide chains linked together by a disulfide bridge. They will bind to fragments of antigens that will be present on some type of host cell, either the antigen presenting cells or the MHC1 bearing cells. In doing so, they will go through and communicate what compounds those cells have interacted with. They're going to be found on both cytotoxic T cells and the helper T cells. Each of the two protein subunits has a constant region, and then we'll also have a variable region that will differ between each T cell. Those variable regions will be specific for a specific antigen in terms of their binding pattern. Once activated, the helper T cells, which are lymphocytes that carry both the T cell receptor and the CD4 receptor, will act to stimulate the activity and growth of other B cells and T cells. That CD4 receptor will work alongside the T cell receptor and act as a way of ensuring recognition. The killer T cells, as we said, are going to carry the CD8 receptor. That CD8 receptor will be located right next to the T cell receptor and will again work alongside that T receptor to initiate recognition of the antigen. The next thing we want to talk about is antigen presentation. Antigen presentation occurs when the pathogen that infects the body is engulfed by one of the macrophages. Those macrophages will then use enzymes from the lysosome to cleave that pathogen into small fragments. Those antigens are then bound to class II MHCs molecules and are displayed on the surface of the macrophage. So as that macrophage goes through and consumes one of those pathogens, a lysosome will be used to break it apart, and then the contents of that vesicle are expelled from the cell. Those contents that have been expelled can then become antigens that will be recognized by B cells. In addition, you have those MHC2 molecules that will have pieces of that bacteria or virus on them. Those virus or bacterial fragments, called antigens, can then be displayed to helper T cells. The macrophage will depart from the site of infection and will move through the lymphatic system to the lymph nodes. There it will encounter helper T cells. It will then present that MHC2 with the antigen to those helper T cells. As the macrophage presents its MHC2 to the helper T cell will then release cytokines to then proliferate its own growth. The activation of helper T cells initiates the acquired immune response. But that is the topic of the next video. So until then, stay safe, stay healthy, and I'll see you later. Yeah. Ready to go. All right.